Hi, this is Mrs. Murdock, and today we are going to talk about how water moves. All right, you don't have to write this, it's already on your paper. How does water move or respond to different gradients? Okay, we're going to make sure we define each term as we go here. So, first of all, again, this is already there. You don't have to write this. Osmosis. All right, the definition of osmosis is a process where water diffuses from, hold on, just give me a second, from where it is high concentration to where it is low concentration. Okay, I know it's off. Just give me a second. The lamp just died. Nope, it's dead.
CPR. I know CPR.
Help, Mr. Bouchard, help. Nope, the video's still going. Hold on. Okay, so now we have our overhead back. So what I'm going to do now is I'm going to draw the same three beakers down here. Just go ahead and copy that, and I'm going to draw the beakers here that you've already got, so you don't have to draw them. There we go. There we go. Okay, all right. All right, so we have these three beakers. I'm even going to letter them just so that we can refer to them a little bit more easily as we go through, okay? And in each beaker, we've got a solution of water and a giant red blood cell. We're just going to pretend that these circles are giant red blood cells, even though we know red blood cells are not that big. This helps us get an understanding of what's going on inside them compared to what's going on outside them. So the first beaker, what I'm going to do, and you can do this with me, has a solution with lots of salt part ions in it. There's lots of salt ions here. So we're going to draw some dots to indicate very salty water that this red blood cell is floating in. And I'm going to say it's 0.8 molar salts. To give it a number. There you go. I can see you all. I can hear you all putting your dots in. That's terrific. All right. Okay. So we have very salty water here. And inside the cell, it's different. Inside the cell, it's 0.3 molar salts, which is less than outside. So we'll just put a few dots in here, not very many. So that's the first beaker. The second beaker, inside the cell is the same. 0.3 molar again. So just a few dots. And outside the cell is also 0.3 molar. So the dots here are going to be about the same spread out as inside the cell. Just a few dots out there, right? So that's beaker B. Beaker C, again, the inside of the cell is still 0.3 molar. We're going to keep that the same. And then outside the cell, there's no salt at all. 0 molar, no salt. So there's some inside, but not outside. Okay, so now let's look carefully at these three situations. So here's what we do. What we do is we have to understand the term gradient we learned already, right? So the word gradient really just means difference. So which ones of these beakers, A, B, or C, is there a difference between the inside and outside of the cells? A, definitely. There's much more salt outside than there is inside. So there's a gradient across the membrane. And whenever there's a gradient, usually something's going to move. Yes? Well, if you can't see, please feel free to go closer, okay? So there's a gradient, and a gradient is what's going to make things move, right? What about the other two? Is there a gradient in B? No, it's the same. So there's no gradient here at all. Okay. What about C? Yeah, there's a difference here too. So there's a gradient in A and in C, but not in B. Now there's another way to describe this. We're going to give a label to each one of these solutions that are outside the cell, a special term that you'll see. You'll see in the textbook, you'll see in the Ed Puzzle tonight, and you'll see on the SOL exam. Yes? You are right. You are ahead of me, Lauren. Very good. The first one, this solution outside the cell is hypertonic to the inside of the cell. Hyper. That means it has more. It has more solutes. Or like what you drew, the little dots. There's more little dots in that solution than on the inside. Than the inside of the cell has. There. Okay. So hyper, more, right? The next one has exactly the same amount, and we call that isotonic. We call that an isotonic solution, is any solution that has the same amount of solutes. As the inside of the cell. Okay. The last one, since there are less solutes outside than inside the cell, we call that hypotonic. That's a solution that has less solutes than the inside of the cell. There. All right, so now that we have that set up and we have these different situations in each beaker set up, right? Now what you need to know is that living cells, this membrane here that's around this cell, is just like the membranes that you've been coloring in and labeling all week, right? Membranes have different transport proteins in them to help things go across them or go through them. So all living cells actually have more aquaporin proteins than almost any other type of channel protein in their membranes. What's an aquaporin? It's a channel just for water, exactly. Okay, so aquaporins are channels for water, and in the membranes of these red blood cells, and in any living cell, in fact, there's lots of aquaporins because water is such an important substance to bring inside cells. And cells can get very sick very fast if they either have too much or too little water. So aquaporins mean that a lot of times when you have these situations, the water is going to move much faster than anything else. And the point of water movement is usually to help the situation reach equilibrium. What's equilibrium? It's balance, reaching equality. Right. So right now, beaker A is way out of balance. So where will the water move, either into or out of the cell, to reach equilibrium in this situation? Very good. You guys are so smart. Yes, the water will move out because there's the white space inside is what the water is, right? So if it's not the dots that are going to move, the salt isn't going to move, the water is going to move to equilibrate the situation. Okay, so as a result of water moving out, what do you think is going to happen to a cell in salt water? It'll die because what will happen to it? Physically, what will happen to the cell if it's losing water? What will it look like?
Raisin.
It'll shrink like a raisin, exactly. Okay, so what happens? The result is water leaves the cell and shrinks. That usually results in it dying, right? Animal cells don't like that. Animal cells, that's a bad situation. Plants too. Plants don't like it either. Okay, next one. In the isotonic situation, what's going to happen? It's going to stay the same, but is water still moving? It's just moving equally both ways. And we kind of draw a double arrow for that, like that. So water moves equally both ways. Equally. And this is called dynamic equilibrium. Because you have a balance, but it's dynamic because the water is still going to move randomly both ways. And animal cells really like this. That's their happy place, is in an isotonic solution. Okay. Move my camera up a little bit. There we go. Okay. What about the last one? Which way will the water move here? Yeah, the only one left is for the water to move in, right? To try to dilute the particles inside. But if it's a pure thing of water, what's probably going to happen to this cell? Yes, now you've got a cell that's really, especially an animal cell, will be at risk for bursting. So water moves in, moves into cell, and cell may burst. Not really apoptosis. Apoptosis is something sort of different. Apoptosis is where the lysosomes inside a cell are programmed to kill the cell, right? That's a little bit different. This is just lysing and bursting because of environmental, yes, which is that's an interesting connection that you made there. Okay, so may burst. Animals don't like this. This is not a happy place for animals, right? Okay, so I do want to add a little bit more information here before I run out of time. Plant cells are a little bit different. Plant cells, remember, have a cell wall, right? That animal cells don't have, with the membrane just inside it. No cells like salty environments. Plant cells will die if they're in salty environments, so they don't like it either. But plant cells actually, they're fine with isotonic solutions, but their happiest situation is here. Plant cells love hypotonic solutions, which is kind of weird. So let's think about that. You've got cell walls, right? And the membrane's filling up with water. So why would a plant cell be okay with that? All that water pressure pressing against that cell wall is actually their way of being able to stand upright without bones, without a skeleton, right? So when your mother waters the plants on the windowsill when they're wilted, they'll perk right up because all of their big vacuoles inside them are filling up with water and lots of pressure and pushing against a million cell walls. That's the way a plant stays upright. So that's actually a great place for a plant. Plants love hypotonic solutions. Animals don't, though, because we don't have those cell walls to keep us from bursting. Okay, and that is all I wanted to tell you about osmosis, and I hope that's helpful.
All right, you guys need to get this.